because it gives me opportunity to show you what we are actually doing in Karolinska. But to tell you what we're doing in Karolinska, I have to start the journey a little bit earlier in Pittsburgh, because that's in Pittsburgh where everything started. Okay, these are usual slides to say what I'm saying is my own responsibility. So we started everything in Pittsburgh. So we are liver people. We work in cell-based therapy. So what we're doing is treating liver disease, transplanting cells, not organ cells. That was done initially in 1992 by two groups at the same time. One was my mentor, Dr. Strom, who did in 1992 with allogeneic hepatocyte. At the same time, without even discussing before together, Dr. Mito did it in Japan with autologous human hepatocytes. So what happened? What we have done during the past more than 25 years, we're transplanting a little bit more than 150 patients, not only us, of course, different centers in the world, including London. We did a lot of them. So we transplanted patients with metabolic liver disease, different kind, and now in the next slide, I'm going to show you the list of all the inborn errors of the metabolic defect we're correcting right now, and we have corrected in the past. But also we rescue patients with fulminant hepatic failure. We treated also patients with cirrhosis, but as you can see, there are very few because they don't respond really well to this kind of therapy because the organ is unfortunately so messed up that even all the therapy we have right now, they are not so efficient despite the fact we are working on that. And of course, in several cases, we transplanted the patient, but the patient was stable, but the surgeon decided when the liver was available, okay, let's go for a solid organ transplantation, since that's the established therapy. So even the patient was quite fine, they decided to go with the liver. And in both therapy, we have immunosuppression. These are all the metabolic liver disease we and the other people transplanted so far. As you can see, many of them are essentially Crigler-Najjar and urea cycle defect. These are the two kind of metabolic defect. Most of the time we address with our therapy for several reasons, because they are quite severe, because many of them cannot even bridge to the organ transplant in several situations, or because the quality of life is quite poor, but even the solid organ transplantation might be quite not efficient. I would like to bring your attention also on the phenylketonuria. We transplanted 2 patients with phenylketonuria so far, actually. One we transplanted in Pittsburgh, and one was transplanted in Belgium by Dr. Sokol. Interesting, both of them have been using a technique that we developed in Pittsburgh, and I'm going to briefly show you in the next few slides, that was quite interesting because it was an allogenic transplantation using cells coming from another defect. So essentially, in our case, we transplanted hepatocyte collected from an MSUD patient and transplanted into a PKU. And the lady now is perfectly fine, of course, under immunosuppression because that's allogenic, but she's totally fine. She's a normal diet. She's quite happy. She's actually super happy right now. So why we like the cell more than the organ? Not because I want to steal patient from the surgeon. Absolutely not. Solid organ transplantation is a fantastic technique. 50 years of them prove it. But what we have is an advantage for the cells. Because when we transplant a cell, as you saw probably in the previous slide, it's a minor surgery. We transplanted the cell in our situation going through the umbilical vein. Our surgeon can easily, he said it's quite easy to re-cannulate, even in quite patient with adult patient as well. And so we inject the cell. And this cell can be injected several times, multiple times. That's what we did actually. Repeated injection is actually the best. The second part is that the organ is still there. So what does it mean? If for any reason my therapy doesn't work, the patient is exactly the same position he was before because he's able to receive an organ donor, an organ as he was before, nothing. And that's what happens sometimes. That's why I mentioned in some situation was a bridge because the patient was better or was quite normal, but definitely the surgeon decided, okay, the organ is the best. Now we have a perfect matching, let's go with the organ. And we did it. But that's a massive surgery, and you can do only twice probably the organ transplantation. With the cell, you can do it every time. You can even boost it. You can do it several times during the year. Because why the cells are interesting? Because for several diseases, metabolic diseases are the proof, you don't need to transplant. You don't need to restore the whole liver. 5% of your liver mass is enough to correct most of the disease. Phenylketonuria, urea cycle. With Crigler-Najjar, we need sometimes a little bit more, but less than 10. So we need to inject the cell. The only problem is, how can we have the cell? The cells are isolated from liver. They are not used for transplantation. And believe me, when a surgeon decide not to use an organ, it's definitely a bad organ. So I've been working for a while in America, as so liver that I cannot even describe. steatotic like crazy, huge, massive of fat with some cell contained. And that was very complicated to rescue those cells, but we did it. So during the past 20 years, which are the main problem we had to solve, that most of them we solved it already. So we had to adapt the surgery practice because it depends on which kind of organ you receive, all organ, a lobe, just a part of that resection. We need to do it in GMP condition because the GMP is essential now. It's like a drug. So the cell need to be done in GMP. We need to select enzyme, which is collagenase, which most of the time was not good enough to go through the FDA. That's why we spent several years to develop with the bioengineering company the perfect enzyme. And we need to prove that we can perform injection multiple times because we inject several cells. We inject billions of human hepatocytes in a patient. And you cannot inject billions of cells in a patient within 1 day or two. We need weeks. Sometimes we need months. Sometimes we need different donor, of course, because they cannot really be cryopreserved. That's a major problem with the hepatocytes. Hepatocytes don't like to be cryopreserved. They are big cells, highly functional. They don't survive really well to the cryogenic. And so the only problem we haven't solved so far is where we get the cell. So far, we get the cell from everything we could, from a fetal liver to a resective part to the whole organ, which can be very large. But every time I isolate the cell, we prove that this cell can be used, running them immediately within a few hours before the surgeon injecting them to prove they are functional. The last thing I want is to inject cells that are not functional in a patient who needs a specific function. For example, a urea cycle defect. I don't want to inject cells that are not capable to metabolize ammonia. There's no sense for me because I'm not going to rescue the patient. I'm not going to provide the function is missing. The other functions are perfectly fine. I don't want to provide the other function with my cell. I need to provide the function is missing. That's why we tested several functions, all the one we can, within 1 hour or two before the surgeon is ready to go, because we need to match the donor and the recipient. Okay, as I mentioned, the problem is where we get the cell. So these are the 5 sources of cell we tested so far. I'm going to have one slide for every source, just going very quickly, because my aim is to show you the latest results on the stem cell, as the title said. So the PKU patient, as I mentioned, both of them were transplanting using this technique. So we isolate hepatocytes from an organ that was explanted. So an organ was considered useless because going to the garbage, what you want to replace. And from this organ, we rescue the cell. We isolate the cell, and we rescue all of them. So we find out that these cells not only are great in viability, so the first column is the organ donor we usually use in clinical. All the disease we tested, all the organ we use, different urea cycle, Krieger-Najjar, MSUD were fantastic cells. That's why we use for PKU. All of these cells, once we isolate from the organ, which is looking very bad, cirrhotic most of the time, they recover because you extrapolate, you move it outside from the background, which is not very supporting, and you put it in the different background. Even when you put in culture, they recover. They're perfect for drug testing. And these are fantastic because we can use it for Krieger-Najjar or urea cycle defects. Of course, if I'm transplanting a Krieger-Najjar, I need to be certain. If I'm isolating the cell, I don't want to transplant him back in a Krieger-Najjar. So the phase 2 is missing, but it's perfectly fine for urea, and vice versa. So the urea cycle is absent because they don't have it, because that's the deficient, but they're totally normal for all the other 300 different functions, hepatic function. So that's why we use it. And very interesting, as a side effect, we tested this cell, we transplanted in a very peculiar model that my mentor, Dr. Strom, developed. It's a quadruple knockout. Essentially, it's a lectityrosine. It's an FAH deficient mouse. We transplanted a human cell. And what we find out that what we develop is new animal model, because I get the cell from a specific disease, from a specific patient. I can generate hundreds of mice with almost 199% of the hepatocytes are human. So the drugs can be tested on patient own cells. Of course, it requires a little bit of time. You cannot do it when it's a risk of life. But that's what we're doing now. We create more and more animal models. They are more similar to human because these are real human cells into a mouse. And how we estimate the repopulation before sacrifice the animal? Wonderful. We just measured the human albumin in the serum. So 1 milligram per ml of human albumin, I know it's pretty much 20% of the population. That's why for me it's easier. Then when we sacrifice the animal, we find out that it was completely repopulated, but only with the hepatocytes, nothing else. Not the immunocells, not the cholangiocyte, nothing else. Only the hepatocytes are human. Then those kind of source of cell was not considered as good by the FDA because, okay, there's some benefits, but there is always some problem because how can you be certain you don't transfer the disease to another patient? We know that because we transplant only 5%. If 5% of the cell is not enough to induce the disease. So if I transplanted 5% of MSUD, you don't get MSUD. That's why domino liver, so the solid organ transplant, has been done for decades. But okay, let's start investigating other cells because we are really keen on that. So we checked neonatal. So unfortunately, several patients, several babies, they died few hours, few days after born. So we try, okay, those organs, we might rescue them because those are the cells that might be a little bit closer to progenitor. And we isolate the cell, and as we were expecting, we find out that these cells are fantastic in viability when they're not very ischemic. Because in one case, for example, the mother wants to hold the baby for 12, 14 hours. Of course, 12 hours later was too late for us to rescue the cells. But most of the time, even after a few hours of ischemia, we get decent cells. And these cells, on the contrary of adult, since are more immature, they can be easily cryopreserved. So we can freeze them and keep it there when we need it for acute liver failure or for a patient coming. But as I mentioned, we don't look only to the viability. We don't care about that. We care, but we care even more about the function. We are liver people. We want to know if our cell perform the function we want. That's why we tested for several ones. As you can see, there are some are very good ones and some are not. That's why our screening is very important to better match the recipient and the donor. That approach was actually so good that another group in Spain decided to transplant the animal for the Krieger-Najjar, the gun rats, and they proved that it was actually working. And not only, these cells are working even in my mouse model. So now I have several mice with human cell, with neonatal human cell, growing and becoming mature. But neonatal are not so many. During the past 2 years in Sweden, I got only 5 of them. So I started testing, especially when I was in America, a lot of abortion. So when we got the abortion, I decided, okay, let's check those fetal cells. Those fetal cells are definitely less mature. I'm going back, one step back. So these are definitely progenitor. What we find out that they are definitely immature. So the viability is great. I'll not even bother you with that because the off-which they're super great. But the problem is they're very mature. So cytochrome P450, 3A4 is the most common one. Everybody knows that, right? In the fetal life, the fetal isoform is what is expressed. It's supposed to switch when you get born. So the first 2 or 3 years of life, that's when you're switching. So you're moving, that's you see the other bar. So they're very low and they're getting very high when you are adults. So there is kind of switch. Unfortunately, this cell requires a little bit longer time to reach a maturation level that we need to correct several diseases, including Krieger-Najjar urea cycle defects. As you can see, when we transplant in the animal, we reach a very nice adult level, but it requires 6 months in the animal. 6 months is quite a long time, especially for some patients they can wait, but some of them, they don't really want to wait so long. So that's why we decided, okay, let's go for the kind of cell that everybody loves right now. They got even the Nobel Award in Stockholm a few years ago for the IPS. IPS, fantastic technique. We started generating tons of them. Starting from human hepatocyte, we generate IPS, but are the IPS, as well as embryonic stem cell, because we tested in the past as well, are they capable to mature into real hepatocytes? What we did, based on our experience, that we're moving from the fetal to adult, young and adult, we have the largest cohort of patients, largest cohort of cells that everybody has ever had. So that's why we are quite strong in this. We start testing our cell. And not only we test our IPS, we went there and we asked to all the company, okay, send me your best cell. I don't want to even know how you did it. I don't care. It's your patent. I don't care. Give me the best cell you have right now, the one you think you are able to transplant the patient to correct the disease. So what we did, we tested all of them. So the first are fetal, the second one are adult. As you can see, my cell, their cell, they are not mature enough. Most of the time, they're closer to the fetal than to the adult, as proved by 3A4 and 3A7 switch. Some genes are better. That's why we're trying to understand now why some of them are getting maturation, which is better than others. And we are working now with the technology that you might have heard about, the organoidal, so in suspension created this kind of organoids. But none of them, neither the best company right now, can produce real mature hepatocyte. That's why we have some problem. The pH for phenylketonuria is quite good, actually. It's not so bad. One company actually was quite bad. I'm not going to tell you which one, but definitely one of them was not so efficient in that. But we are reaching a kind of nice level. The problem with IPS, so far, the risk of creating a tumor is still present. So that's why there is a lot of problem before going to clinical. We tested several genes, not only this one. I'm not going to bother you with another 60 genes because you guys, I don't want to, you're getting bored and you're falling asleep. Because I want to go to this one, to the stem cell that we are actually moving to the clinic now. So during all the testing we have done, the best tissue I ever found, and believe me, we work hard with our team, we test everything, was the amnion membrane. What is the amnion membrane? So this is a placenta, full term, after normal delivery. The amnion is this kind of tissue that I'm holding on my hands. Why is it so important? Okay, first of all, placenta for me is fantastic because there are plenty of them, more than I can process. The baby get delivered every day, no problem for me during the daytime, so it's saving also my marriage because I don't have to work at night. Fantastic. Even in Sweden, when they get only 17% of cesarean section. Why cesarean section? Because for sterility reason. To go to GMP, I need to prove that I'm sterile from the very beginning. So normal delivery sometimes is not very clean. So that's why we're going for that. And Sweden is not the best one. I mean, London, UK is better. Italy, where I'm from, is much better. They have even more C-section. But believe me, 70% of the birth is more than enough, so I don't have even time to work on all of them. And what is the amnion membrane? It's not the amniotic fluid. I want to clarify this. It's not, of course, cord blood either. It's the kind of tissue that is surrounding the baby. It's a very peculiar tissue because there is no vessel. There's no blood in that. It's a very thick membrane with few stromal cells embedded, very few, and this kind of epithelial cell. The baby is on this side, so they're facing the baby. They're protecting. These are actually the cells producing the amniotic fluid, in large part of that. Part of the amniotic fluid is coming from the fetus as well. But they're the one involved in the delivery. That's why the breaking water, these are the cells causing that. These are the cells, more important, protecting the baby from the mother. Because we need to remember that the baby is 50% maternal and 50% paternal, or sometimes even completely allogenic. So I spent several times to develop a technique that will allow me to collect only those cells, not the stromal one, not because I don't like them, but because the stromal one, I think they're very useful for several diseases, probably not liver one, because they're not solid proof that they can really mature into hepatocytes. And these cells are very interesting because I had to go back and study a little bit more about embryology now because I find out that these cells are actually very, very, very interesting cells because they originated even before the 3-germ layer are specified. So they're in the blastocyst at the beginning of the second week. These are the very first cells the embryo needs to create to protect itself from the mother and to attach to the uterus to get the blood supply. Otherwise, the baby is going to be immediately discarded. So these cells are very peculiar. So they originated from the epiblast even before the 3-germ layer we're going to give born to the fetus. Instead, the mesenchymal one, into the amnion membrane, they're coming from the mesoderm. So they're not originated at that time. That's why I don't want the MSC. I want only the epithelial one. And why the epithelial? Because these epithelial share a lot of characteristics with the pluripotent stem cell. They have the same nuclear and surface marker that we measure on embryonic and IPS. They have it as well. They can differentiate in all 3-germ layers. We prove it. We are liver experts, so I can tell you how far they're going through the hepatocyte. I don't want to discuss about the pancreas or the brain. That's some other people's jobs. They are not tumorigenic. We already transplanted in patients. We transplanted in animals. Never got rejected. Never formed any tumor. They're not immortal. They don't have telomerase. That's why they don't grow like crazy. I'm not expecting this cell to repopulate the whole liver. These cells are very interesting for me because once I put into the parenchyma, and this is a kind of picture that this is mouse liver, and these are my human cell. It's very easy for me to find the human cell in the mouse. So I find out that these cells communicate, have a lot of paracrine effect with the surrounding environment. Not only as a soluble molecule, but they secrete a huge amount of vesicle, the secretom that everybody loves right now. So they secrete a plethora of cytokines, of metalloprotease, of enzymes. We are still investigating to find out why and they keep secreting this. Probably during the pregnancy ever since, after we isolate, they might be useful, but they might stop doing some of them. But some of them, they're not. Second part, when I injected my human stem cell into a mouse, I don't need to make them hepatocyte before injecting. I put it there, and the liver of the mouse communicate with them. And within a few days, they become completely mature. When I'm talking about mature, and once again, I'm comparing my cell, that's the second bar, to fetal, or the black bar is adult. So as I pointed out, that the white bar is my cell before injection, when I isolate it. If I put in the liver parenchyma, they become mature. For most of the gene, so 23 over 24, the only one is UGT1A1, which is not super high. It's quite close, better than fetal, but it's quite close. So many of them, they are expressed as adult form. But it's only gene expression for transporter, cytochrome P450, and all the enzyme for different metabolic disease which are expressed, or is also the enzyme working? So which is the best way to test it? Get the animal with the human disease and test this cell. So that's what we did. We started in Pittsburgh. At that time, in Pittsburgh, we had a quite large cohort of MSUD patients, with all the Amish community. We got a lot of patients who got transplanted for MSUD, so we were interested in testing. So we started transplanting human amnion epithelial cells, so placenta stem cell, into a mouse, an MSUD mouse, which I want to underline that is absolutely immunocompetent, non-immunosuppressed. So the first thing is to immediately recognize my cell because it's a xenotransplant and reject it. What we find out that is not only not rejecting them, but it is rescue most of the animals. So 82% survival. The animal usually is dying in less than 1 month if you don't do anything because it's as an MSUD. If I'm transplanting my cells, 82% of the animals survive. What happened to the remaining 18%? I cannot tell you because we never found the body. Unfortunately, the mother has this kind of bad habit to kill the baby when he finds out that he's affected, so we don't know if he was able to survive or not. Just simply the body disappeared, the mother got it. It's a kind of cannibalism that we cannot do anything for, but definitely they survive. The growth curve, so the body weight is perfectly fine. It's quite similar to the normal wild, to the wild type. How many cells? So getting the human into the mouse parenchyma is very easy. I just need to check for a human DNA. I find out that a few days after injection, actually 3 weeks after injection, I got around 4% of my cells. 4 weeks later, a little bit more. So these cells proliferate, not like crazy, but they proliferate. What about the activity? Because that's what I want, the enzymatic activity to correct the MSUD. This animal model, the MSUD mouse model, is actually not perfect because there is still 5% remaining activity in the kidney and so on, which is not sufficient for life, so they die, as I mentioned. When I give 5% of AE cells, I got pretty much 5% of increase of activity. That's an indirect proof that all my cells become mature, so mature that they can express the enzyme that I really want to. We check the serum and the brain for different amino acids. As you can see, the black bar is the affected mouse, the sick mouse, and the wild type. As you can see, several situations improve. In some cases, completely normal. So when I'm going to 100 days later, so 3 months later, that we stopped the study because we moved from Pittsburgh to Karolinska, we find out that several amino acids are improved, highly improved, if not completely normalized. We check with other people also the neurotransmitter, and we find out even in this case we got a serial improvement. That's why we think we can even correct the neurological problem in these patients. But MSUD was not enough. So PKU is another disease I like because I was working in Pittsburgh once again, and the National PKU Alliance, the PKU Foundation in America, they really want us to find a cure. We offered them the hepatocyte transplant, and they were quite happy, but they're not super happy because we want something more efficient, with more cells, with less immunosuppression. So we said, okay, we're going to test these cells, which are quite promising. We transplanted several fusions, as we usually do in clinical, with different placentas, so because these are different donors, allogenically, immunocompetent, no immunosuppression. Once again, these cells, they didn't get rejected, neither recognized. If I'm transplanting syngeneic hepatocytes, so mouse hepatocytes into a mouse, the improvement is this blue bar. Both in the serum, then in the brain, phenylalanine decreased. If I'm injecting the human placenta stem cell, the improvement is much higher. And it's very surprising that it is in the brain, it was completely normal. In serum, we still have a little bit higher level. But the brain was the first one to be corrected. I was expecting exactly the opposite. That's why we're working out to figure it out why it's possible. Of course, I detected how much DNA, and once again, we got animal between 5, in one case we got 15%, so it was quite a lucky shot. The cell don't express pH. Few weeks after I put in the liver parenchyma, they're expressing. And they correlate really well with the DNA, a little bit, even too well. So, but is pH the only enzyme expressed when they become mature? Absolutely not. We check also in the mouse parenchyma for PKU, all the other different enzymes, 3A4, the 1A2, the different CYPs, but we check also the other metabolic ones, CPS1, alpha-1 antitrypsin. In several cases, they are similar to adult or quite close. Alpha-1 antitrypsin is one of the lowest ones, but we are very close to the adult one. Of course, in the brain, once again, these are the human cells, these are syngeneic mouse hepatocytes, which are supposed to work better and better tolerated. They're not. The human cells work even better. The monoamine pathway, I'm not an expert, but I was quite surprised as well about it when I saw this data because they're very nice. 2 metabolic diseases I show you. I want to briefly show you this because it's still ongoing. We are working with Toshio Miki in University of California. The OTC, urea cycle defect, is one of the major causes of patients we lose after birth, so we want to transplant them. Immunocompetent mouse, we inject a couple of doses of cells. As you can see, we got a 50% reduction in orotic acid. That's what you measure. And they can tolerate the ammonia challenge, so they can really metabolize ammonia. Working with another person in Italy, where I'm from, we got in contact with that guy for this kind of disease that I never heard about before, or at least I heard about, but I wasn't an expert about. So the complement factor H, which is a kidney problem caused by deficiency into the liver. That's why when I face him, I said, probably I can treat that because it's a missing enzyme in the liver. It's a missing protein. Then I can produce with my cell, and I'm going to probably rescue your animal. And that's what happened. One injection of the placenta stem cell, we got a highly less deposition of that complement in the kidney glomerulus, which caused survival of the animal. The animal never developed the disease. That's why now is even more interesting. He was doing this study with MSC, now he wants more of my cells. Then, since we are quite a crazy people, we had a surgeon, we didn't know which kind of crazy project to give it to him, we decided, okay, let's try to move aside. So not metabolic liver disease. Let's try something even riskier, the acute liver failure. Another cohort of patients we transplant with hepatocyte. So if I give a little dose of D-galactosamine to my mouse, can I rescue in some part, or can I help him to regenerate if I give him my stem cell from the placenta? So when we decide to give it, we set up a dose that's killing 100% of the animal within 3 days. We decided that was kind of perfect range because in human it's like 1 week. So if you're going overdose, you have the time to recognize and go to the ER, whatever. So all the animals died. If I give 1 dose of my cell, all the animals survived. Absolutely normal. What I find out that 2 days later, the transaminase drop. Several pro-inflammatory cytokines drop as well at 2 days and 14 days. So it was a strong regenerative potential, a strong regenerative effect driven by this molecule, the cytokine IL-10. Another molecule that I never paid so much attention because it's not so common to get. I find that is actually very, very important because that's one of the most important key molecules for regeneration and immunomodulation in general. So we start to investigate this immunomodulation. How is it possible that my stem cell from human, they don't get recognized by the immune system in the mouse? How they can avoid that? And why, if they can do that, they don't form any tumor? So more important, when I started reading the paper, I realized that several women, when they're pregnant and they have an autoimmune disorder, the autoimmune disorder reverts. And once they deliver the baby, they go back to the original status. So there is something going on during the pregnancy who's helping them to the autoimmune disorder. Of course, quite easy to understand that I thought it was my cells. So we start investigating the cell, and the placenta stem cell, the amnion epithelia cell, only that cells from that amnion tissue, amnion membrane, express and produce a lot of anti-inflammatory immunomodulatory molecules. The same one identified before in mesenchymal stroma cell. And those are the same molecules probably involved in the graft versus host disease treatment. They're not highly immunogenic. They don't express class 2, they express very low class 1, and they express class 1b, another molecule that I didn't know anything about, so I need to go back to my university book and start studying back all the immunogenicity. So I found out that class 1b is actually the one you like when you want to tolerate the organ when you transplant it. And my cell express a very high level, exactly like class 1. No, it's class 2. They express a lot of HLA-G, unfortunately not HLA-E. When you want to do everything at the greatest part. So I say, I was quite disappointed. I say, this is another class 1b that I would like to get expressed. I found out they express by insoluble form as much as HLA-G. That's why they create, they are not only protected, but they create a kind of protecting environment. And what happened to my vesicle that I mentioned before, they produce a lot of vesicle. The vesicle they produce, they're very different from MSC. They have all the epithelial marker, but they express a huge amount of HLA-G as well. So even the vesicle can be used as a drug to treat. Okay, let's go a little bit faster in this part because, of course, when I start to notice one molecule instead of the other one, I decide to spend money and say, okay, let's screen all the surface molecules in these cells. These cells are definitely new, are definitely different. I'm not going to show you 332 graphs. I'm just going to show you a few of them. Why? Because I need, and I really need, because reviewers are always jumping to my throat for this. They are not MSC. All the MSC markers are negative. They have a lot of markers in common with pluripotent stem cells, but I don't dare to call them pluripotent. For me, are multipotent, which is for me is more than enough. There is only one molecule, CD73, which is very highly expressed in MSC. And every time I got a review, so they express 73, so they are MSC. So no, the others are all negative. Why they are expressing? So I start working with other people, they are more expert than me. So the CD73, together with other molecules, are ectonucleotidases, are surface enzyme which metabolize adenosine and create this kind of immunotolerant environment. MSC most of the time require T cell to complete this kind of circle. My cell express both of them, 39 and 73. Not only, there is a guy in Turin, University of Turin in Italy, who identified an alternative pathway, starting instead of from ITP, from NADP, and I gave him my cell to him, and find out that all my cells expressing all these ectonucleotides as well. So this is the very first cell we ever measured that express all the machinery for all of them. That's why they have such a strong immunomodulatory potential. It's so important because when I expose my cell to the T cell, the proliferation of the T cell is suppressed. So the T cell don't proliferate. The NK don't proliferate. The B cell proliferate. And that for a certain time was very scaring me. And then I'm going to show you the last slide in 2 seconds, show you why at the end I was relieved from that. We find out another molecule that we are studying right now, CD47, which is protecting my cell from macrophages, from the apoptosis, and the complement 55, 59. I don't show you too much, it's too much, but these are also ongoing. So these are all the molecules that my cell express without manipulating, which protecting them from all the immunocells. So far we identified these kind of 4 important molecular pathways, which are probably the key molecule to allow my cell to escape recognition. They don't cause tumor, but they just are protecting them from the immune system. So what's happening when my cell, they're cuboidal, they're epithelial, get in contact with B cell or T cell or NK or macrophages? Essentially, this is what's happening. The T cell, the few T cells survive are the T regulatory cell, pro-regenerative. The B cell I show you proliferating. I was scared at the beginning. Then we find out the old B cell become B reg. Very nice, pro-regenerative. The macrophages switch from M1 to M2, pro-regeneration once again. And then NK decrease. So all the immunocell seem to cooperate with my cell in order to regenerate more than to cause a chronic inflammation. And that's where we are right now. So most of the problem we solved how to isolate the cell, we identified the cell, we proved they are safe, because as I mentioned, animal and human has been transplanted, they never got any problem. We are in GMP, so we develop a technique to isolate and to create a GMP batch, so we are quite safe for that's why we got the permission to transplant patient. We correct so far 4 metabolic liver disease, and we rescue animal for acute liver failure. And thanks to the all immunomodulatory molecule we identified so far, and the many more that we might identify in the future, we propose to transplant this allogenic stem cell in human without immunosuppression for the very first time. And we got permission for. So that's where we are right now in Karolinska. So we got the permission, we are in GMP creating the first cell bank, because I need probably several cells to perform different patients. And we are identifying the patient. We have permission to transplant the first 10. We don't know which disease. PKU people in America, they're quite aggressive in that, but even MSUD and all the other diseases we correct are probably the best candidate right now. The urea cycle defect is another one I would like to transplant, but that's where we are right now. So in Karolinska, but essentially in the world, because there are no other people doing. I need to point it out that this was done thanks to Christian Zworak, who was excellent when we were in Pittsburgh. Then both me and my mentor, we moved to this side of the ocean. Now we are in Karolinska, and we put together another team of people in Europe, which is supporting us very, very efficiently, and that's why we are running very fast in this. I have to thank absolutely the PKU people for sponsoring me, for giving me the money to do most of the transplant, because otherwise I couldn't do that. And thank you all for the attention.